Today I'll be describing our invention of nanobugs and specifically how it could be used to combat climate change. But before I dive too deep into how nanobugs are made, how it could address the problem at scale, I thought I would describe the problem itself. So the amount of greenhouse gases, here specifically carbon dioxide, has been at the highest level at 408 ppm parts per million right now, which has been the highest in the last 3 million years. And 17 of the 18 warmest years on record have occurred since 2000, and more than 800 million people, or about 11% of the world's population, is now vulnerable to climate change. So perhaps we are doing something to try and address this. Well, not quite, because in 2018, that's when we have the recorded data, the global carbon emissions were at an all-time high of 37.1 billion tons of carbon dioxide that was emitted. So where does this carbon dioxide go? How do we breathe? Well, there was a lot of media reports recently when there was a fire in Amazon, and that's good because 1 in 4 breaths that we take come in from the rainforest, but the remaining 3 of the 4 breaths, or 70% of Earth's oxygen that we breathe, comes in from the oceans. So perhaps we are taking better care of our oceans. Well, it turns out again not quite, because we dump massive amounts of plastic into the oceans, and by 2050, if the current trend continued, we would have more plastic by weight than fish in the ocean. And again, this problem is unimpeded because we produce close to 380 million tons of plastic waste each year, a large majority of which ends up in the ocean. While it may seem like catastrophic, almost cataclysmic problems, but herein we could use this crisis as an opportunity and see if both of these could be combated simultaneously. And I will describe how this could be done through nanobugs. But before we do that, I thought I would just describe how big the scale of the problem is. So just to give you some sense of how much plastic weighs, the 380 million tons, well, how much is 380 million tons in number of people? And you're welcome to choose one of the 4 options. And it turns out that it's equal to the weight of almost the entire human population. And the way I came to this conclusion is, let's assume an average human weight of 75 kilograms. And again, babies don't weigh that much. And so even with that assumption, you would be accounting for almost 5.1 billion people, or 2/3 of the entire human population. So the problem is big, and it really does need to be combated at scale. And the only way this could be done is through use of solar power, which has been the primary source of energy on our planet. And there are different ways in which solar power can be harnessed. One is through inorganic photocatalysts, and these can easily absorb a lot of light. They conduct charges pretty well. These inorganic materials end up on our rooftops as solar cells. However, when you try to make chemicals out of them, you kind of get a mixture of products, making the process more expensive. And it's also very limited to a small range of products like hydrogen and some low carbon materials. Well, plants do this in a very intricate manner, where they produce biomass, and we can either pre-treat or hydrolyze this biomass or undergo fermentation to make some highly specific and complex chemicals. However, plants don't absorb light that well. They have very limited absorption, and the process here is pretty lengthy, going from sunlight to the fuel or the chemical that you're looking for. So the overall efficiency of converting incident light or energy from the sun to the specific chemical is fairly low efficiency. So we have low selectivity on inorganic materials and low efficiency on the organic materials. So perhaps the solution is some hybrid of this biological and this man-made inorganic material process, and these are our nanobugs. And these nanobugs can potentially combine the best of both by having strong light absorption, and it can generate charges pretty efficiently and can make highly specific and complex chemicals very selectively. So they can combine high efficiency with high selectivity. So how do we make nanobugs? Well, we've created this process, so it's incredibly simple now. And this is what the process looks like. We take the bugs from soil that we would like, and we simply add these quantum dots or nanomaterials that have been engineered with specific layers, and we'll talk about these layers. Once these quantum dots are added, they get taken up or gobbled down by the cell. Then they go in and attach to these specific proteins or enzymes, and this very specific chemistry is what drives the sunlight driven biocatalytic process or conversion of carbon dioxide or even air into specific chemicals that we want. So this entire process can be highly efficient and lead to specific generation of highly complex chemicals. And we can do a whole range of reactions and make a number of products almost like a factory. So what are the challenges? Why don't we just go ahead and do this? Well, there were several challenges that we have encountered and solved over the years, as it's documented here. Specifically, first we have to teach the cells how to eat quantum dots. This is not something that they do by themselves. And then there is this very specific enzyme nanomaterial coupling that needs to be engineered. We have to teach cells then how to eat light. And then we have to ensure that these nanomaterials are non-toxic, that the cells can grow with this, and a whole range of balancing issues that would ensure efficient conversion of incident sunlight into specific chemical fuel. So there's a whole range of living nanobugs that we've created, and the first one that I'm going to talk about are these quantum dots and Azotobacter vanillandi. These are just bacteria that live in the soil and convert massive amounts of nitrogen into ammonium salts and ultimately proteins. And this entire process is done on a very specific protein known as molybdenum iron nitrogenase. And as I said, we've isolated now bacteria that live in soil and attached quantum dots to those specific enzymes that are required for this biocatalytic transformation. And now it can directly use this nitrogen gas from air. So we don't really need to supply pure nitrogen, and it can fix this in an air environment. Nominally, any trace amounts of oxygen, even parts per million of oxygen, would completely deactivate this enzyme, requiring a fairly complex and air-free condition. But these bugs can maintain air-free conditions. So we need living nanobugs, not just any combination of enzymes and quantum dots. So how do we engineer these quantum dots? So we start with the core material, the inner layer. So we take a whole range of quantum dots here. I'll show you 3 different colors of each. So let's say we chose these 3 quantum dots with varying sizes. Just by adding a couple of layers of atoms on its surface, we can change its color all through the blue, visible, and then near infrared or region where we cannot even see. So it absorbs sunlight very efficiently. And then we can test whether these quantum dots can now excite these specific enzymes and cause these nanobugs to carry out the catalysis or conversion of sunlight into complex chemicals that we desire. And it turns out that our measurements correspond very well with how efficient nanobugs are from each one of these quantum dots, and only quantum dots that have a specific quantum state or energy level over the energy level of the enzyme can inject their charges, because as we know, whenever light falls into these inorganic materials or semiconductors, it generates electric current, and that electric current only can drive this complex chemical formation process. So the second step is to ensure that the quantum dots attach very selectively to the enzyme that we want and nowhere else. So in order to ensure this coupling, we had to go through some design steps. For example, here in this conversion of ammonia, and just for calibration, this ammonia formation process or Haber-Bosch process is arguably one of the most important industrial processes. We use about 4% of all our energy into making ammonia and ultimately fertilizers. So it's an industrially important chemical. And when we attach these designed quantum dots to this specific, either we take the cell and we lyse it completely, or we just take this purified enzyme, you can see very clearly that not all chemicals. So we started with cadmium sulfide, and then by comparison I'm showing you zinc sulfide. If cadmium sulfide quantum dots are directly added, they attach almost everywhere. This is the cell lysate, which has all these different enzymes. This is the enzyme that is required, and cadmium sulfide is virtually attaching everywhere. But the zinc sulfide material is selectively attaching on this enzyme that we want. So what we do is we start with a core material that matches the energy level of the enzyme, that can excite this enzyme and carry out the catalysis, and then we cover it with a very thin, almost an atomic monolayer of the material that would allow this selective coupling. And we can show here that you can then make a whole range of inner core materials. Here I show you two different core materials in green and yellow, and either you look at the entire lysate from a nanobug, or you take specific enzyme and you mix quantum dots with it. You could see that this fluorescence or bright emission coming in from the quantum dots corresponds very well with just the enzyme, and it's not attaching anywhere in the cell lysate. So the process is very specific, but more interestingly, when we coat this very thin layer of zinc sulfide, it massively increases this efficiency of biocatalytic chemical or fuel generation. And the black curves here show you what happens if we just add the core quantum dot. And then when we add the thin one monolayer or two monolayer zinc sulfide shell, you could see this massive increase in the generation of that specific chemical or fuel. So let's see, how much quantum dots are we talking about when we say let's make a mole of nanobug, and say if a nanobug has 10,000 copies of an enzyme. In that tiny little bug we have 10,000 of these enzymes. How much quantum dots would be required to make 1 mole of nanobugs? And it turns out the answer is 1200 kilograms, because for every mole of quantum dot, or every mole of nanobug, you would require 10,000 moles of quantum dot, every quantum dot for every enzyme copy. And using a quantum dot molecular weight of about 120 grams per mole, you would require around 1200 kilograms of quantum dots for 1 mole of enzyme. And later on we will see how much material can this 1 mole of nanobug make. So then we had to tune how much of this thin layer of shell material do we want to put. And there was, of course, a detailed investigation in this. As you can see, the core material has a lot of defects on its surface, which leads to this broad emission. But then when we grow a thicker and thicker shell, the number of defects on the surface go away, which could be good news. But you cannot do this indefinitely, because as I'm showing using very detailed electrochemical measurements, that as we grow thicker and thicker shell, the charges, the positive and negative charges that we generate in this core material have a long way to make through to the enzyme which is attached on the surface. So what we found was 2 atomic layers or 2 monolayers of this shell material was enough and actually optimal for this biocatalytic conversion. So we chose this as our material or thickness of choice. So now we have a core material and then we have a shell material, and you could see that we could carry out very efficient conversion or biocatalysis through this process. We could just take water and make massive amounts of hydrogen, and this process is driven just by this very specific enzyme quantum dot chemistry. So once we kill the nanobug and we lyse the cell, just to show you this, at physiological pH, everything works really well, but we could sort of decouple the quantum dots in case, we were worried about, well, maybe something's happening in there. Maybe the quantum dots are in there, but they're not really coupled to the enzyme. When we decouple the enzyme or the protein with the quantum dot by adding a chemical called imidazole, you would see that all this catalytic reaction goes away, which is very significant. Similarly, when you increase the pH, you add more acid again, here the bug is dead and we are just trying to decouple the enzyme with the quantum dot. You could see by simply changing the pH by just 2 points here, deprotonates or protonates the histidine and decouples the enzyme with the quantum dot, and again this catalytic conversion goes away. And please remember this chemistry, because we could definitely use this to decouple the enzyme and make fresh batch of quantum dots and recycle our quantum dots with new batches of bugs and make new generation of nanobugs. So how does this light-driven or sunlight-driven process work? We simply take water and air, and that's it. And we add this to the nanobugs, and you can see massive amounts of hydrogen and ammonia that are formed, which is the industrially important process that gets used up. Now, before all of this gets done, we have to ensure, or we have to teach the bugs to eat the quantum dots. They nominally don't eat the quantum dots. So just to demonstrate how this engineering or this nano engineering works, we coated these quantum dots with different tiny little molecules. Here, molecules which were fairly short, had a negative charge, and we saw very modest uptake, less than 10% uptake, which wasn't optimal. When we keep the molecule pretty much the same, but we replace it with a positive charge, we see a huge increase, almost 40% uptake. So it may seem like the bacteria are gobbling it up, but what really happens, and I'll show it to you in a minute, is that these positively charged quantum dots go and attach everywhere on the bacteria, the lipids, the proteins, the DNA, RNA, and end up killing the bacteria, which is not what we want. So the solution really was to use zwitterionic material, the ones that have both a positive and a negative charge on the surface, so they don't really attach non-selectively to any of the cell organelles. And these are essentially amino acids, essential amino acids. So it also makes it easier for us to teach bacteria how to eat it. And the uptake is pretty modest. 10 to 15% of all quantum dots end up inside the bacteria attached to the enzymes, which is very powerful. Now, after the bacteria eat it, we have to make sure that they are living and they're viable and they're happy. So with the negatively charged molecule that I showed, you would see the bacteria growth inhibition. So bacteria are not really growing that well, especially when we add more and more quantum dots, close to 1 micromolar quantum dots. So there's some toxicity. But when we look at these essential amino acids, these zwitterionic or positive and negatively charged surfaces, these were non-toxic. The bacteria were growing just as they would without the quantum dots. And remember, I said when we put positive charges, it just ends up killing the bacteria. And here's the evidence that even at fairly low concentration, virtually nothing, you could see a huge decrease in the bacterial growth. And it's not just the growth, it's also the viability of the bacteria. So the cell viability is measured through its metabolism. And we see here that only with these essential amino acid coated quantum dots, the bacteria are fully viable at 100% or so. And with these positive charges, the bacteria are not viable. Their metabolism is down to 20% or even lower, which is really bad news. And when we test essentially colonies of these nanobugs, we again see what we've seen with the other tests, that positive charged quantum dots are not good, but only these zwitterionic quantum dots can give you good quality living viable nanobugs. So once we form these nanobugs, let's see if they can actually catalyze the specific chemistry we were looking for. And again, with the negative, zwitterionic, and positively charged quantum dots, we see what mimics essentially the viability of the bugs. Because if the bugs, as you remember, are not living, the oxygen from the atmosphere can get in and deactivate the enzyme, and it's not going to be able to make any ammonia. And that's reflected here in this turnover number, TON, which is a measure of how good or how efficient the catalyst is. So for every mole of the cell, one single cell can give you 30 million moles of ammonia here, which is massive in just one single run of about 3 hours. So it's just generating massive amounts of ammonia, which is not true for the negative and the positive charge, because the cells are not viable and the oxygen gets in. And even though you have these enzyme quantum dot hybrids, they get deactivated and the cells are not living. And only with these essential amino acid or zwitterionic coated quantum dots, we have these massive turnover numbers or massive product generation. We can now expand this to a whole range of different quantum dots, all the way from ultraviolet to visible to near infrared or infrared region of the sun. So we can virtually capture all facets of sunlight as shown here with different quantum dots. And that's really good news. But let's see if this would lead to product generation. And actually, that's exactly what happens. We see similarly high, tens of millions of moles of ammonia produced in just a matter of few hours with all of these quantum dots. And this variation in their turnover number or catalytic yield is just a measure of how much light they're absorbing and not any real consequence of the material itself. So now we've actually created these nanobug factories. So if you look here, there's a very well-known 2-to-1 ratio of hydrogen to ammonia that gets formed when you simply take air and water, and we do see that in just a matter of 1 to 3 hours. You will see that the bacteria are producing almost 30 to 40 million moles of ammonia per mole of cell, which is extremely rapid at a rate that's unprecedented. This is the turnover frequency. How quickly can nitrogen from air absorb or go inside this cell and get converted into ammonia? And almost 10,000 ammonia molecules coming in per second, and almost 5,000 hydrogen molecules coming out of the cell per second, which is massive. When you look at the efficiency of this conversion, how much sunlight can end up as energy into this fuel or the chemical, this quantum yield or quantum efficiency per quanta or per mole of photons, how much of this energy ends up in the fuel. It's close to 13%, which is much like solar cells. So solar cells will just give you electricity, and there's obviously the problem of what to do when sun is not there. But here you're making fuels or chemicals. So you could make all the fuel that you want during the day or when the sun is there, and store it and use it whenever you'd like, at night, days when it's cloudy, and so on and so forth. So there are massive, not only could it do it at a massive scale, but also at efficiency that meets or could even surpass solar cells. And here's how you can actually make nanobugs that are better than solar cells. So when we turned up the light intensity, what we see is that the quantum efficiency or the quantum yield here goes down. And you may wonder why. I'm giving it more light. Why is it not producing more and more chemical or fuel? That's because now we've made these nanobugs so efficient that they absorb the sunlight, they absorb massive amounts of sunlight, and now they're really waiting for these enzymes or proteins to finish making this chemical. So in a way, now there's so much energy being transferred to the enzyme that they are taking their time, and they are the rate limiting or the limiting factor in this conversion. And due to this mismatch between sunlight, which is nominally called diffuse, or people say, well, sunlight is weak. Well, not weak enough for this, because with sunlight, which is extremely high intensity for these nanobugs, the maximum theoretical yield can only be 16 to 20%. But now we have 13% efficiency, which is extremely high, and points to the fact that if we can increase this maximum theoretical yield by adding more nanobugs, so the sunlight can be absorbed with more nanobugs, then you can push up this maximum theoretical yield and make more and more and more of the chemical in these factories. And just to show you that it's not just ammonia, what we did was we made a whole range of chemicals. Fuels, isopropanol and butanediol, these are gasoline additives. You can directly make them and add them to the tank of your car. methyl ketones, which is biodiesel. You have ripening agents. You have bioplastic, massive amounts of bioplastics being produced, again, at a rate that's about 1% or so, with different quantum dots, all the way from ultraviolet to near infrared. And again, this is primarily limited by the mismatch between how fast this enzyme can do chemistry versus how intense the sunlight is. And if you compare it to, another evidence that this is all limited by the biology now, which could be tweaked, by the way. You could take bacteria from the soil and treat it and create synthetic microbes that could have many, many more copies of the enzymes. So for the same 1 mole of cell, you could have 6 to 8 times more fuel or more chemical, driving up this efficiency from 13% all the way to perhaps 60-80% conversion, which would be incredible and useful, as I will describe in a minute. But here, the nanobugs surpass the natural yield. So in the natural yield, you could try and do this catalysis by giving glucose, massive amounts of glucose without sunlight. And even with this natural yield, you get about 150 to 200% conversion with just a saturated bioreactor. So it's working better than the best bioreactors and that very controlled environment. And this process can be scaled up very efficiently. So we went from tiny little cultures to a lab scale up, or lab scale up. And we could make gram scale of this biodegradable plastic, which was great. In just one run, we could make, several grams of PHP. And we can make now tandem cells. So different colors of sunlight are utilized by using different quantum dots, as I showed, to make a specific fuel and really create like a factory. And you can change how much of these is made by changing how much light is being absorbed through each one of these quantum dots. And you could make more biodegradable plastic or more biodiesel, depending on the demand and seasonal usage and how much fuel you have at the end of the day. So these factories are amazing, and just to show you that you can, after you're done, you've extracted your product, you could simply take the nanobugs and put them in a fresh solution that has no product, or just remove the product, and you can just cycle them day and night, on and on. So this is just 3 cycles, so several days worth of data, and you get pretty much flat turnover number. It's not really degrading. The enzymes are great. But here, when you look at the turnover number into making a whole range of fuels, ammonia, ripening agents, biodegradable plastic, again, hundreds of million moles of biodegradable plastic that could be made, which is massive, by just 1 mole of cells, it could really attack this problem of climate change at scale. So let's see, if we convert all of the CO2 that we emit in a year, 37.1 billion tons in 2018, and use nanobugs, which have a turnover number of, say, 7 times 10 power 10 moles of PHB per mole of cell per year, how much volume would you need to use up all the CO2? Would you need a small reservoir or a pond, a large lake? So this is, remember, this is no mitigation steps. Just business as usual, and all of the CO2 could be converted into massive amounts of plastic or biodegradable plastic, which would solve the problem of plastic pollution too. Would you need a whole sea full of them or an entire ocean? So let's see if we can do this at scale. And the answer is, all you need is a large lake. All you need here, and I walk you through these calculations, that all you really need is just 1.7 kilometer cubes of these cells, which is approximately a large lake. And I just gave you some specific examples from places I've lived in and places where I live. For example, Navajo River Reservoir here in Colorado, or Lake Minnetonka in Minneapolis. And it could sink the entire CO2 for the entire world. But of course, you don't want to do this at a single location. You want to spread this through tiny little ditches or ponds all across the globe, so you could convert CO2 into biodegradable plastic and just use that and not make any more plastic that lives for centuries in oceans. So I hope I've convinced you that nanobugs are something that could be a scalable alternative or solution to this big problem of both climate change or emission of greenhouse gases and generating massive amounts of biodegradable plastic at scale. They have an efficiency like solar scale. They're extremely scalable. All you need to do is make them in bigger and bigger volumes. It's an extremely low cost fuel, and you can make a whole range of specific chemicals from fertilizers, drop in fuels, to bioplastics, to preservatives and ripening agents, all of which we've made now using different colors of sunlight. It's a fairly easy to implement low tech technology. If you can brew beer at your home, you can definitely use and generate nanobugs. And it's fairly sustainable to make nanobug factories. So I'd like to end by thanking all my grad students and postdocs, and specifically Yuchen Ding and John Bertram, who actually did all of the work, and the funding agencies, specifically DOE, that really made this work possible.